Welcome to our channel. Let's dive right into a mystery that's probably keeping you up at night. You put in the work, you grow a flawless crop, you visually verify that those trichomes are perfectly mature, and you make the cut. But then the lab results come back, and your yields and potency are completely unpredictable. Why is that?
Seriously, why do two plants with identical trichomes under a jeweler's loupe test entirely differently at the lab? It's an incredibly frustrating problem, and it actually drives this controversial practice of lab shopping just to find the cannabinoid concentrations you're hoping for. But here's the thing, the labs aren't broken. It's our primary method of visual inspection that is totally failing us.
And the stakes for this guessing game are staggering. When you rely on subjective visual cues to trigger a whole plant harvest, you're looking at a devastating 26 to 46% crop loss. Think about that. You are literally sacrificing massive amounts of developing seed and resin, all based on what essentially amounts to a gut feeling.
To understand why this happens, we really need to separate visual maturity from chemical maturity. For decades, we've treated clear, milky, and amber trichomes like they're some exact map of the plant's chemistry. But the hard truth, that visual assessment is entirely disconnected from the objective, hidden chemical reality.
The science on this is actually pretty clear. Human eyes, even when we're squinting through microscopes, just cannot perform reliable chemical analysis. We're essentially trying to read a highly complex biochemical textbook simply by judging the color of its cover.
So what is actually happening when a trichome turns amber? Cultivation lore has always told us that amber equals overripe, degraded THC, right? It's a foundational industry myth, but we have to ask, what is actually accumulating inside that resin head?
Enter flavonols. These are plant compounds, like quercetin and camphorol. As a plant ages, it actively pumps these flavonols into the plant's cuticle and the trichome to provide UV protection and structural rigidity. They're totally normal and essential, but they possess a unique optical property that is completely tricking our eyes.
We call this the amber illusion. That brown or amber tint you're seeing, it is not degraded cannabinoids. It's actually a heavy accumulation of flavonols, acting like a tinted window. They emit a green-yellow fluorescence that completely masks the fact that the cannabinoids inside are actually degrading.
Please like and subscribe, it really helps our channel a lot. So, if flavonols are basically pulling a fast one on our eyes, how do we look past that tinted window? How do we see the actual invisible chemical markers of peak maturity in real time without having to destroy the plant in a lab?
The answer actually lies in light. Specifically, deep ultraviolet light at exactly 280 nanometers. Neutral cannabinoids like THC and CBD have a very unique excitation matrix. When you hit them with this specific UV light, they fire back a distinct, measurable fluorescence.
And the data on this is fascinating. As the flowering period progresses, the intensity of this specific trichome fluorescence consistently increases across different strains. It gives us a direct, measurable, optical readout of the plant's true chemical maturity, completely bypassing that tricky amber illusion.
We can literally map these optical signatures. Under targeted UV, clear trichomes emit a weak blue ring around the secretory cells. Chemically mature, milky trichomes blast an intense, uniform blue right through the cavity. And those overripe amber trichomes, they redshift into a dim green-yellow emission. So how do we actually deploy this commercially? Enter LIFE, which stands for Laser-Induced Fluorescence Endoscopy. It's a cutting-edge agrophotonic tool that uses monochromatic laser excitation to probe the exact biochemical state of intact living plant tissues right there on the stalk.
Now, operating sensitive optics in a blazingly bright commercial greenhouse is tough, but LIFE overcomes all that ambient sunlight by firing ultra-short pulsed UV lasers, like a 350 nanometer xenon flash. This is paired with synchronized time-gated cameras that physically block out the sun, capturing only those nanosecond fluorescent photon emissions. And just to be clear, this is strictly a botanical application. Unlike medical endoscopes used in gastrology that might rely on injected dyes, this agricultural life system relies entirely on measuring the plant's natural autofluorescence to track secondary metabolites. There are no dyes, no cutting, just light. Here is what the continuous flow of the future automated harvest looks like. First, a flexible life endoscope scans the apical buds. Next, an AI instantly classifies the trichome maturity. Finally, automated tools selectively harvest only the flowers that have reached their absolute peak chemical maturity.
This completely unlocks the harvest top buds first paradigm.
By only taking those perfectly ripe apical inflorescences, you expose the lower canopy to a flood of light and nutrients. This allows those lower, shaded buds the time they need to mature further and reach their own peak potency.
Executing this kind of precision requires some seriously advanced robotics. We're talking articulated arms designed to avoid rigid obstacles, targeted panicle mowers, and even 15-watt, 405-nanometer blue-purple precision cutting lasers. They cleanly execute the harvest without damaging the remaining plant structure at all. By embracing the selective, technology-driven approach, cultivators are leaving the old whole plant harvest in the dust. That massive 26 to 46% loss of seed and resin we talked about, it is entirely eliminated when you harvest based on precise, individual flower maturity.
Contrast this in situ harvesting with traditional post-harvest mechanical processing. Usually, harvested plants are hit with liquid nitrogen, dropping them to an extreme minus 45 degrees Celsius or minus 49 degrees Fahrenheit. This embrittles the stalks to mechanically snap the trichomes off. While cryogenic freezing is absolutely fantastic for post-harvest extraction, it doesn't solve your timing problem. Life-guided selective harvesting ensures that those immature lower flowers are never cut down prematurely in the first place, guaranteeing maximum value when they are eventually cryogenically processed. The efficiency numbers with this new paradigm are just stunning. AI-driven optical scanning and selective robotic harvesting can reduce crop loss to negligible levels. We are talking under 2 kilograms per hectare, which is less than 1.8 pounds per acre. It's an unbelievable optimization.
We are witnessing a monumental shift in cultivation. We're finally moving away from subjective bro-science visual assessments and leveraging objective, science-backed precision agriculture. We are quite literally farming the invisible chemical reality of the plant.
So the real question is, will you continue trusting your eyes, or is it time to trust the light? Thanks for watching. This content does not constitute medical advice, legal advice, or recommendations for the use, self-treatment, or consumption of any substance.